So good evening. I bid to Dr. Gita and my fellow friend. My name is Nur Izzatur Syamira binti Baharum. My metric number is 197435. And today I would like to present the case study. So the first case study is about genetic mapping. And this example I will use the genetic mapping for the fruit fly. So as you can see, to calculate, we can use RFLP to calculate the genetic distance between two loci. So, but this technique can be used when we are able to observe recombination. So, as you can see, in the colorful pictures I provide at the left of the slide, the pink one is indicate the female fruit fly, while the blue one indicate the male fruit fly. So they are basically four different RFLP bands on the female fruit fly, but there are only two RFLP bands on the male fruit fly. So this is because the female fruit fly is heterozygous, while the male fruit fly is homozygous. So this is further simplified into the southern blot for RFLPs. As you can see, male fruit fly have two different bands, which is 1 and 2, while female fruit fly have 1, 3, 2, and 4 bands of 4 RFLP. So the females can produce 4 different gametes, but male can only produce one type of gamete, which is 1 and 2. Two other possible 4 are called parental, because they carry both RFLP band from the same chromosome, which is 1 and 2 from the left and 3 and 4 from the right of the chromosome. The other two chromosomes are recombinant because their recombination have occurred between the two loci, and thus the RFLP bands are mixed. So now 1 is now linked to 4 and 3 is linked to 2. So when these two flies meet, the frequency of the 4 possible progeny can be measured. And from this information, the genetic distance between the two RFLP loci, upper and below, can be determined. So as you can see, 35% and 35% of the offspring are produced from the parental genotypes X, while 30% were from the 15% and 50% are produced by recombinant genotype X. Thus, we can conclude that the two RFLP loci are 30 centimorgans apart from each other. So next, I will present the case of screening for the sickle cell gene. So the sickle cell genes are genetic disorder in which both genes in the patient include the amino acid valine. So as you can see here, beta A is the normal. Beta A is the normal individual, while beta S is the mutated one. So as you can see in the second of the genes in the codon 6, instead of we having A, which GAG that encode for glutamic acid, will have valine we have T that GTG that encode for valine. This will cause the sickle cell disease. So the only differences are the substitution of T instead of A. So these small substitutions will convert a GAG codons to GTG, and this also abolish the CTGAGG to CTGTGG, which spans codon 5, 6, and 7 that are recognized and cut by one of the restriction enzyme. So when the normal normal gene, which is beta A, is digested with the enzyme, and the fragment separated by electrophoresis, the probe bind to the short fragment, which you can see as the yellow mark between the red arrow. But when we have a substitution of T, so the probe cannot bind to the sequence anymore, so it will produce a longer fragment, which you can see between the blue arrow. As you can see on the left is the plant is a family, is a pedigree of a family whose they produce a infected child. So this infected child is a son. At first, the both the mother and the father are heterozygous. So they will produce a son that is homozygous and is infected with the sickle cell disease. As you can see, below is the electrophoresis of the family. As you can see, there are two bands, beta S and beta A, for the heterozygous individuals, while for children 1 and 3, you can see only one band because they are homozygous. But they have more prominent bands because they have twice as much as DNA in them. So in this example, a change of a single nucleotide produced by the RFLP. This is a very common cause of RFLP, and cause, this polymorphism are now commonly called as SNPs or SNP. But not all SNP, not all RFLP arise from the SNP. So how can this tool be used? By testing the DNA of prospective parents, their genotype and their genotype can and the odds of having afflicted children can be determined. So in the case of a sickle cell disease, if both parents heterozygous, so there will be a 1 in 4 chance that they will produce a child with the disease. So by using amniocentesis or chorionic villus sampling, they can determine if they can choose whether they want to abort their children or bring the afflicted children into the world. Okay, there are basically three problems arises from this technique, which is the mutation that cause most human genetic diseases are more varied than the single mutation associated with sickle cell disease. So, such as the cystic fibrosis genes, there are more over 1,000 different mutations that can cause the disease. So, we need more than one probe that can identify the disease, but the using of more than one probe will cause false negative, which that people that do not carry this gene are falsely accused. So next is there are many diseases which result from several mutant genes working together to produce the disease phenotype. And there are still genetic disease for which no gene has yet been identified. So we need more effort for the gene to be located, clone and sequence. Until the genes can be located, clone and sequence, no probe can be made to detect it directly. So that's all from me. Thank you. Stop share.